All right, it's currently 3 p.m. here at Natural Partners in our Scottsdale, Arizona location, and it's time to get started. Just so everyone knows, we will be doing a full Q&A session at the end of the presentation. You can submit your questions in the chat window at any time during the presentation, and we will gather them all and answer as many as we can at the end. If we do not get to all the questions, we will follow up with you individually to make sure the questions do get answered. We'll also be making this presentation available to everyone who registered for the webinar. The link will be provided in our follow-up email within a few days of this presentation. And with that, I think we can begin. If you have not heard, your presenter today is Liz Roth-Johnson, PhD, who is a part of the scientific affairs team at the science-based nutraceutical and reference standards company Chromadex. She received her PhD in molecular biology from UCLA and her bachelor's degrees in molecular and cell biology from UC Berkeley. Thanks for joining us today, Liz.
Great, thank you so much. It's a pleasure to be here. So today we're going to try to cover kind of a lot of science about NAD and NR quite quickly. So here are our learning objectives for today's webinar. First, we're going to spend some time thinking about NAD, what it is, why it's important, and different factors that can affect NAD homeostasis. We're then going to look at a strategy to increase NAD using a molecule called nicotinamide riboside, or NR for short, and explore some of the preclinical and clinical evidence around boosting NAD with NR. So before I jump into the science of NAD, I want to start with a little bit of historical context for this molecule and the scientific community's interest in it. The story of NAD really began more than a century ago when scientists first described its importance in the process of fermentation. Its connection to human health really became a lot clearer in the early 1900s when the vitamin deficiency disease pellagra was first characterized. Vitamin B3 was born when forms of this vitamin were shown to cure pellagra, first in dogs and then in humans. These vitamins, we now know, are used by cells to create more NAD. So really, pellagra is the result of cells not having enough NAD, and it's associated with some pretty nasty side effects, including dermatitis and dementia, and can ultimately lead to death. So this was really the first clue that NAD is a really important molecule for human health. Over the next several decades, scientists established NAD's role in cellular metabolism and energy production. We'll get more into the details of that, but during this time, NAD was largely thought to be conserved in these processes. So in the 1960s, there was a big discovery that led to a paradigm shift in the NAD science field. This discovery was the identification of NAD consuming enzymes. And so these are molecules that actually use up and consume NAD to carry out their processes. And so this really set the stage to further research that led to sort of the recent resurgence in NAD research and renewed interest in studying NAD as a means to support human health and healthy aging that we're now seeing today. Okay, so what is NAD and what does it do? Classically, NAD has been described as a redox cofactor based on its established role in oxidation reduction reactions in all living cells. Redox reactions generally are reactions that involve the transfer of high energy electrons from one molecule to another. And so NAD can facilitate these reactions by accepting electrons from one molecule and donating them to another. So what we're looking at on this slide is NAD's role as a redox cofactor in cellular energy production. So during the process of cellular respiration, which you may remember back from an early biology class or biochemistry course, NAD in its form NAD+, accepts high energy electrons from food molecules. When it's holding on to these electrons, we call NAD NADH, and that NADH can donate electrons to the mitochondria to drive production of the energy storage molecule ATP. So another way I like to think about this is through an analogy, and I think this might also be helpful if you're ever trying to explain this to some of your patients. I like to think of the cell as a light bulb, something that requires energy to function, to be powered. And then we can think about food, our energy source, like a battery. If you put a battery next to a light bulb, nothing's going to happen. You need to connect those two things with a wire, with a circuit. And really that's what NAD is doing. It is providing a connection between an energy source, our food, and the cell, the thing that needs that energy, so that energy can be transferred into sort of a usable form. NAD also serves as a redox cofactor in a huge number of metabolic processes in the cell beyond just energy production. I like to consider NAD together with its close relative NADP. These are very similar molecules, and NADP is created in cells from NAD itself. And together, NAD and NADP help cells build nucleic acids, they help synthesize fats, they help make steroid hormones, they're involved in scavenging free radicals, and a lot more. I mean, really, it's kind of hard to find examples of metabolic processes that don't involve these molecules in some capacity. But one thing I really want to reiterate here is that as a redox cofactor, NAD is largely conserved. So you notice in all of these cases, NAD is just shuttling back and forth between its form NAD plus and NADH, right? So it's NAD plus when it's not holding onto electrons, can collect electrons and become NADH. It can then drop off those electrons and convert back to NAD plus, and overall levels are kind of the same. So this was sort of the view of NAD for a long time in its role in redox reactions. But more recently, there's been a lot of interest in a new type of molecule that consumes NAD. These are molecules that actually break NAD into two different pieces in order to carry out their function. So this is where we're really starting to move beyond redox in our NAD story. So there are different molecules called sirtuins, called PARPs, called CD38 that all consume NAD to carry out their cellular functions. If you look at the little diagram on the right, you see the molecule, the molecular structure for NAD. What all of these molecules are doing is they're taking that NAD and they're actually breaking it into two separate pieces here that we're calling ADPR and NAM. And so this is different. NAD here is not acting as a redox factor, it's acting as a substrate that allows these different enzymes to do their jobs. And so this science behind these molecules, these enzymes, is still pretty new and emerging. But generally, we're learning a lot about sirtuins and their role in maintaining cellular health. They can do things like regulate gene expression to promote things like mitochondrial biogenesis and health. PARPs have a pretty well-established role in DNA repair at this point, with some other emerging functions in cellular maintenance. And CD38 has some roles in calcium signaling in the cell. So these touch a lot of different cellular processes and consume NAD along the way. And so the discovery of these molecules really got scientists thinking more about NAD and NAD levels. And so at the same time that we've been trying to understand and characterize these NAD consuming enzymes, we've started to also make more observations about NAD levels and how they are changing in a variety of different conditions. So I know there's a lot going on here on this slide, but basically what this is showing is a summary of different peer-reviewed observations of NAD levels changing in association, and specifically declining in association with a variety of conditions. And so every one of these entries here represents at least one published study in mammals, so either rodents or in humans, where designated with the blue dot, of NAD levels declining. You can see in particular that aging has gotten a lot of attention around declining NAD levels. And so that's also part of what's really led to an interest in understanding NAD and its role in supporting healthy human aging. Okay, so what's actually contributing to changes in NAD levels? Right, we have NAD plus sort of at the center of this diagram, and levels of NAD plus can be influenced by changes in NAD synthesis, right? So how much NAD are we making? Can be influenced by NAD consumption, how much NAD are we consuming or using up by those different enzymes we just saw. It can also be influenced by our redox reactions, right? So if NAD plus is being converted to NADH and tied up as NADH, that affects how different metabolic processes can use these molecules. And all of this is important to think about, in part because NAD plus as a substrate for sirtuins, these NAD consuming molecules we talked about a moment ago, can actually support the healthy function of mitochondria. So there's sort of an emerging idea that having sort of enough NAD plus around helps sirtuins function to help keep mitochondria healthy. And so what we're starting to see more and more is that certain metabolic stresses and aging, things that kind of tax the energy and metabolic systems of cells, can perturb this NAD system. So for example, in aging, observations have been made of NAD synthesis decreasing, so the ability of cells to make more NAD goes down, while NAD consumption tends to increase. So more NAD is being used up, and so that can decrease the overall net amount of NAD plus available in the cell. Certain kinds of metabolic stress can also influence NAD plus levels. Overnutrition is one great example of that, where it can shift NAD synthesis, maybe NAD consumption, but also shift the redox ratio of NAD plus and NADH. So for example, the effect of alcohol consumption in the liver is a really good example of this. NAD is used in the first two biochemical steps in the liver to detoxify alcohol. And chronic alcohol consumption has a kind of well-known effect on the liver, shifting what we call the redox state, where a lot of NAD plus accumulates as NADH, leaving very little NAD plus behind. And that can have a lot of widespread effects on other metabolic processes, like burning of fats or production of energy throughout the rest of that tissue. These changes in NAD plus levels also then can have downstream effects on things like sirtuin activity and mitochondrial health. And so actually a number of research groups have now hypothesized that declining NAD levels may actually be a driver of decreased sirtuin activity and mitochondrial dysfunction. So sort of with aging, two really classic observations that have been made at this point are that NAD levels tend to decline with age, and mitochondrial dysfunction tends to increase with age. It's sort of seen as a hallmark of aging. And so I think we're still working out exactly the mechanism there, but more and more scientists are starting to see NAD plus levels as a driver for mitochondrial function and dysfunction. So we're going to get a little bit more into so the preclinical evidence around boosting NAD and what that can do. But first, I just want to make sure we spend a little bit of time talking about how cells make NAD. So NAD is synthesized by from molecules that are known as NAD precursors, molecules that have the potential to be turned into NAD. And these are typically derived from our diet. So the classic NAD precursors are the first vitamin B3s that were discovered back in the early 1900s when we were figuring out how to cure pellagra. And these are nicotinic acid and nicotinamide. The amino acid tryptophan can also be used to make NAD through a much more complicated and drawn out process. So although all of these molecules can be used by cells to make NAD, they do come with some limitations. So for example, nicotinic acid, which is also commonly known as niacin, activates a particular cellular receptor, a G protein coupled receptor called GPR109A. And activation of that receptor leads to an uncomfortable flushing response. So trying to take niacin or nicotinic acid in kind of higher therapeutic doses can lead to this uncomfortable side effect. Nicotinamide has a sort of interesting biochemical side effect in that it actually can inhibit the activity of sirtuins. So this is kind of an interesting side of nicotinamide, because even though it can increase NAD levels, which are then used to help sirtuins function properly, nicotinamide itself can then inhibit the very sirtuins that we might be trying to activate by increasing NAD levels. Tryptophan also comes with some downsides because the pathway, the de novo pathway that uses tryptophan to make NAD involves so many different steps. And many of the intermediates in this process are shuttled off into other metabolic processes. This is a very inefficient pathway. So huge amounts of tryptophan are actually needed to regularly synthesize enough NAD to keep up with cellular demands. One other aspect to all of this I just want to point out is that there are rate limiting enzymes in all of these pathways, right? So all of these pathways converting B3s and tryptophan to NAD involve a number of chemical reactions, each catalyzed by an enzyme. And some of the enzymes involved in these processes have actually been shown to reduce their activity with age or in certain conditions of metabolic stress or disease. So for example, NAMT, which is an enzyme that helps convert nicotinamide to NAD, has actually been shown to decrease its levels and activity with age. So in 2004, a new vitamin B3 sort of hit the scene. There was a discovery in 2004 that certain enzymes in the cell called NRK, NR kinases, could use nicotinamide riboside as an NAD precursor. So this established nicotinamide riboside as a new form of vitamin B3. And this new vitamin B3 has really caught, I think, the attention of the scientific community because it has some unique qualities that set it apart from the existing vitamin B3s and from tryptophan. In particular, it doesn't cause flushing. It does not inhibit sirtuins based on all of the available evidence. It has a quite short two-step pathway, right? So it's a much simpler, more direct route to NAD than either nicotinic acid or tryptophan. And there's even some emerging evidence in a few preclinical studies, and I think more work is needed to really understand this. But there is some emerging evidence that the enzymes that you convert NR into NAD, these NRK enzymes, may actually be activated or turned on in certain conditions of stress. So for example, a study of heart failure in mice showed that in this condition, NRK enzymes were actually upregulated and activated, whereas NAMT, which converts nicotinamide into NAD, was downregulated. So as researchers and scientists have been trying to identify strategies to boost NAD levels, a few other alternatives have been proposed. The idea of supplementing directly with NAD has come up, and also supplementing with the precursor NMN, nicotinamide mononucleotide, which can be converted into NAD by cells, has been proposed. And one thing to consider with these molecules is that unlike the B3 vitamins or tryptophan, these are larger and phosphorylated, negatively charged molecules. And so really what that means is that they require special transporters to be able to enter cells. And so it's currently not clear if if these molecules are really able to easily access cells. There may be specialized cell types that have the right transporters to let these molecules into cells. But to date, there are several published studies showing that both NAD and NMN are actually broken down outside of cells and converted into molecules like NR to then be taken up into cells where they can then be converted into NAD. So let's talk now a little bit more about the emerging science around NR and what that might mean for your patients. So far, there have been more than 100 preclinical studies in cells and in animals that have really helped us understand the science behind NR. And so we'll take a quick look at some of those findings, particularly studies of NR supplementation in rodents. To date, there have been 4 published clinical studies of NR, and there are many more underway. We'll also take a quick peek at some of those studies and think about what might be on the horizon. There's way more research out there than we'll have time to cover today, so I do want to encourage you to check it out for yourself. We have an NAD education hub that's available at practitioner.truniagen.com, where you can find all of the studies that I'm going to mention today, plus many more. So you can hopefully get some ideas from today's presentation. The next several slides are going to give you a quick overview of the kinds of research that's available so that you can then hopefully go and dig more into the specific areas that interest you the most. One thing I want to remind you of and have you keep in mind as we go through the next few slides, kind of summarizing some of the research, is that there's really an overarching theme here that NR boosts NAD to help support mitochondrial health. And so I see that as sort of the underlying mechanism of all of the different physiological outcomes and benefits that we're going to talk about. And because mitochondria and energy production are so important for cellular function, I think this has a lot of implications for human health in a number of tissues and conditions. All right, so first up is NR being studied in the context of metabolic health, again, in preclinical animal studies. So these are all rodent studies, mostly in mice. And the studies listed here on this slide have shown benefits of NR supplementation on metabolic health, particularly NR having protective effects against things like overnutrition. So for example, the data here on the right is taken from this Canto et al. 2012 study. And it shows, among other things, that NR helps to limit gains in fat mass and improves exercise endurance in mice that are being fed a high-fat diet. I'm going to go through these pretty quickly again, just because I want to give you a quick summary and overview, but all of the studies that I'm going to mention on these slides are referenced at the end and found on that NAD Education Hub. Another study looked at NR supplementation in the context of aging. So this study looked at NR supplementation in aged mice and specifically found improvements in mitochondrial function in muscle stem cells. And so as shown in the figures, this had some great effects on the muscle function of these mice. So not only were there increases in numbers of stem cells in the muscles of these aged mice, but their endurance and their grip strength also improved as a result. A few different studies now have examined NR's effect in mouse models of heart failure. This is a really great example of a metabolic stress on a tissue. The failing heart is the heart that has had to work harder than normal to keep up with demands, pumping blood throughout the body. It's trying to generate huge amounts of energy, depends highly on mitochondria, and trying to support mitochondrial health in this tissue could potentially do a lot for the failing heart. And so all of these studies have shown that NR can help preserve cardiac function in these different models of heart failure. I actually want to point out a couple interesting things from the DeGay et al. 2017 study. On the left here, what they found is that NR, but not nicotinamide, one of the other B3 vitamins, was able to increase NAD in the heart. So I just think this highlights some of the distinctions between NR and the other B3 vitamins, and one of the reasons why we want to kind of consider all of these unique NAD precursors individually and not lump them together. Second, this is one of the findings I was mentioning earlier about the pathway involved in converting NR to NAD being upregulated in conditions of stress. NMRK2 is the enzyme responsible for converting NR to NAD. And in this study, they saw that the mouse model of heart failure levels of this enzyme were elevated compared to the control. So it's almost like these cells were kind of looking for NR to support NAD and mitochondrial health. Finally, just to kind of highlight the main physiological outcome here, NR supplementation helped preserve cardiac function, in this case by helping the heart continue to efficiently pump blood throughout the body. Another really big area of research in the NR world is neurology, looking at NR's potential to support nerve health both in the brain and in the peripheral nervous system. Several studies in particular, in this first bullet point, have looked at studying NR in mouse models of Alzheimer's disease. That's an area that's gotten particular interest. And so the data here on the right is taken from the Howe et al. 2018 study. And it shows some results from a Morris water maze test. And so this is a test to look at outcomes related to spatial learning and memory. And in this test, they found that NR had benefits on the performance of mice with basically from 3 different models of Alzheimer's disease. So next to the wild type control, there are 3 different categories. Those are all 3 different models of Alzheimer's disease, and in all 3 cases, NR had beneficial effects on performance in this test. And so one of the really, I think, exciting things about the NAD field and NR is that right now, it's such a hot, exciting topic that new studies are being published all the time. So I just wanted to highlight this one study in particular because it just published yesterday and actually kind of opens the door to a whole new area of NAD research. This study looked at NR supplementation in the context of postpartum. And so they actually fed NR to postpartum rodent mothers. It studied mice and rats and looked at the effects on both the mothers and their offspring. They saw all kinds of changes. A few of the key takeaways were that they saw increased milk production and more time spent nursing. They saw increased micronutrients and bioactive factors in the milk. And they actually followed the offspring who were nursed by NR supplemented mothers into adulthood and saw that they had kind of lasting physical and cognitive benefits compared to the controls. So it's a really interesting study. It'll also be in the NAD Education Hub that you can all check out. So of course, I think the big question with all of these exciting preclinical studies is to what extent will all of this translate to humans? So to date, 4 clinical studies have been published, and in general, these have focused on safety and kind of basic pharmacokinetics, just to see what is NR doing in the body. And these studies show over and over that NR safely and effectively increases NAD levels in human blood and in human blood cells. So far, doses as high as 2,000 milligrams per day have been tested for as long as 12 weeks in typically middle-aged and older adults. 2 studies that were published last year are particularly exciting because they represent the first studies that are beginning to look at NR's effects on health outcomes beyond sort of the initial pharmacokinetic studies. These are small pilot studies. It's important to keep in mind, but I'm going to show you just a couple of highlights from these studies to give you a taste of what has been done, and I think it points to the kinds of work we might expect to see in the future. So last year, a study came out of University of Colorado Boulder by Martens et al. And this study, in addition to being the first study to look at chronic supplementation of nicotinamide riboside, this was looking at 1,000 mg per day for 6 weeks, they began to measure some preliminary outcomes associated with cardiovascular health. And so this is a very small study. It's about 24 people, over the course of 6 weeks. But in this preliminary kind of pilot study, they did see some trends toward decreased arterial stiffness and decreased blood pressure. And so I think that this will hopefully set the stage for follow-up work in a larger cohort to really further explore these outcomes. Last year, another study was published by Dollerup et al. Looking at 2,000 mg per day NR over the course of 12 weeks in older obese men. They looked at a wide variety of metabolic outcomes in these men. And actually, a lot of the results in that study were negative. It was a 12-week study in a small cohort. But they did see an interesting trend toward reductions in liver fat in this study. And so in particular, if they looked at the men in the study who began the study with greater than 5% liver fat content, nearly 70% of the subjects on NR saw a reduction in their liver fat content over the course of 12 weeks, compared to only 39% of the subjects in the placebo. And so again, it's a trend, it's a small study, more work is needed, but I think this sets the stage again for sort of future clinical directions and research. There are currently more than 20 clinical trials registered on clinicaltrials.gov that are not yet published. They're either in preparation for publication or still ongoing and recruiting. And overall, if you kind of take a look at everything that's there, the 3 really major areas of research right now and interest in the clinical arena are sort of neurological studies, looking at how NR can support peripheral nerve health and brain health. Cardiovascular studies, so again, kind of following up on some of the preliminary work that was done by Martens et al. Also looking at trying to translate some of the heart failure results from the preclinical studies into humans, and metabolic health. So looking at, overnutrition, type 2 diabetes, and to see how NR can support health in these areas. Again, I think overall, when I look at this, I see that, NR has a lot of potential to support mitochondrial health. And to me, that is sort of the thread that ties all of this together. All of these different health outcomes are impacted to some capacity by mitochondrial health and the ability of mitochondria to continue to function properly under different types of stress. So again, I invite you to, go to clinicaltrials.gov, look up these studies, see what's on the horizon. I also encourage you to go to the NAD Education Hub and dig into some of the preclinical and the published evidence yourself to see what you think and see what's most of interest to you and your patients. And while we eagerly await for more research on NR to be published, you are able to provide NR to your patients with Tru Niagen. You can find this at naturalpartners.com. If you look up nicotinamide riboside, Niagen, or Tru Niagen, and if you do that, you'll see two forms there. There's a 300 mg per capsule Tru Niagen Pro and a 250 mg per 2 capsules kind of regular Tru Niagen. They both contain the same active ingredient, which is Niagen nicotinamide riboside. This active ingredient has been notified to the FDA both as a new dietary ingredient and as generally recognized as safe. As I mentioned, it has multiple published human clinical trials. All of the published clinical trials of nicotinamide riboside are Niagen nicotinamide riboside. And so we have all of that evidence that NR safely elevates human NAD as an oral vitamin. Tru Niagen Pro is now available specifically to healthcare practitioners. This is a 300 milligram per capsule dose. Our labeling recommends taking 1 capsule daily. That can be with or without food. And really, we recommend any time that's most convenient for your patient so that it's easy for them to remember to take it kind of at the same time of day every day. Tru Niagen Pro is NSF certified for sport. And again, this is really exclusive to healthcare practitioners. And so the larger dose per capsule, we hope gives you the flexibility to work with your patient to figure out what's right for them, kind of based on your evaluation of their needs and the available evidence about NR supplementation. And so again, along with the theme that we've been talking about, Tru Niagen Pro helps to replenish NAD levels, which then leads to all kinds of important cellular health outcomes, including supporting healthy cellular metabolism, promoting cellular repair, so in particular cellular repair processes associated with repairing mitochondria and promoting healthy mitochondrial function, and then ultimately helping to promote healthy aging. So I think at this point, we have some references here, so you can hopefully go find those in the NAD Education Hub at your convenience. But I would love to open it up for questions and have more of a conversation about all of the things you're interested in and hearing more about NAD and NR.
Hi, Liz, thank you so much for the great presentation. I wanted to let everyone know that we also have Bria Ostendorf, who is the Director of Healthcare Outreach, to help answer your questions. So let's go ahead and jump into the Q&A with a quick reminder that if you have any questions, please type them through the chat window. So let's go ahead and answer our first question.
Sure, thank you. This is Bria, and we do have a couple questions from our attendees. The first being, do you foresee any contraindications for the use of NR with active cancer? I'm interested in the supportive and protective effects of NR on myocardial function with regards to doxorubicin use.
Yeah, no, that's a great question. And, I'm going to be honest, it's a tough one to answer. Currently our, clinical studies of NR have been done in healthy adults. So we don't really have any evidence in that particular population. Certainly there's no evidence so far that, NR supplementation would promote the formation of cancer or the development of cancer. But what it's going to do to existing cancer is, I think an open question, just to be perfectly honest with you. And I think one where I would really encourage you to, explore some of the available literature and kind of make, your own expert decision based on what you know about your particular patient and their cancer. Cancer is such a unique kind of disease. I will also say, one of the applications of NR that's currently under clinical investigation is its use as a protectant against chemo-induced peripheral neuropathy. So there are, clinical trials in the works to begin exploring NR in the context of cancer and cancer patients. But at the moment, I think it's one where you'll have to evaluate kind of your particular patient's needs and whether you think this would be a good idea for them.
The next question, and thank you everybody for submitting such great questions. These are questions that we frequently get asked, so I appreciate everybody's engagement. So it's regarding the half-life. Is it better to really divide the doses?
Yeah, no, that's a great point. So in the clinical trials so far that have been conducted, they're actually given twice a day. So our current labeling recommendations suggest once a day, and to be honest, I think that's mostly out of ease of use and compliance. It's easier, I think, generally for patients to remember to take something once a day as opposed to twice a day. But in the last two studies that were published that I mentioned, the Martens and the Dollerup, looking at the higher doses of Niagen, they did split up those doses, once in the morning and once in the evening. So I think there's certainly reason to do that, especially if you're thinking of prescribing or suggesting or recommending a higher dose to your patient.
Okay, we have another one in here, and this is kind of related to dosing as well. Where did Tru Niagen decide on the 300 milligram a day dosing?
Yeah, so the 300 milligram per day is largely based on our non-clinical toxicology studies in rodents, and the sort of new dietary ingredient notifications that came out of that set those serving sizes. So currently our sort of regular Tru Niagen exists in a 250 mg per serving form, or a 300, now actually recently a 300 mg per serving form. And so that's the sort of history of where the 300 came from. The Tru Niagen Pro then puts 300 milligrams into one capsule, just so that you then have more flexibility in terms of how much you want to recommend your patients take.
Yeah, and I will let listeners know too that there are human studies available on that practitioner.truniagen.com, and they have studied multiple doses on there, 100, 300, 1,000.
Right, thank you. That's a great point just to bring up. So yeah, the initial clinical studies did look at single doses of 100, 300, and 1,000. The more recent chronic studies have looked at much higher doses. But actually, if you look at clinicaltrials.gov and see what's kind of on the horizon, there's also studies in the works looking at lower doses, 100, even 300. 300 is a very common one, and then 1,000 as well. So we'll continue to hopefully get more data regarding those doses and what they do to the human body.
So we have a question here, and this is a great one that I hear all the time as we go around the country visiting practitioners and speaking with them. How does a practitioner know if someone's successful on a supplement like NR?
Yeah, it's a really great question, and it's I think it's an interesting one because ultimately, NR is it's a form of vitamin B3. And so really, I think its potential is in kind of supporting and maintaining healthy cells and healthy mitochondria. And so, what a patient feels when they're taking that, I think is going to depend a lot on who that patient is and what their needs are, and sort of why you're, imagining that NR is something, that Niagen is something for them. If somebody's being proactive about their health and healthy aging, they may feel nothing, right? It's like, when you're taking care of your car, you take it in for regular maintenance when it's working fine because you don't want it to break down, right? You don't notice anything until something's going wrong, right? So in that case, you may not feel anything or notice anything. On the other hand, if, a patient is, maybe looking for an energy boost and you think that supporting their cells and mitochondrial health might be good for a particular reason, that might suggest how you might expect them to feel differently or change over time. I will say, just based on customer feedback we've gotten through some surveys, most commonly our consumers report feeling more energy and just kind of overall feeling better, a general sense of better sense of well-being after taking Tru Niagen.
And related to that, Liz, we had a question come in. At what point, is this is NR kind of consistent use, daily use, chronic use, supplement, or at what point could it be discontinued?
Yeah, that's also a great question. We recommend daily use because it is the kind of thing where it's a water soluble vitamin. So you take it, it'll increase NAD. But then if you stop taking it, we would expect NAD levels to go back down. And so really it's through continued use that you elevate and maintain elevated NAD levels. And if you wanted to then discontinue use, there's currently no evidence to suggest any harm in doing so. We would just expect NAD levels to eventually kind of go back down as that source of NAD precursor is no longer being given to the body.
We have a number of questions here that are coming in around testing and laboratory testing to check on improvement, how validating NAD levels. So can you talk a little bit about what is or is not available today?
Oh, like in terms of measuring patient NAD levels, just to clarify? Yeah, so I mean, I know we get that question all the time from our consumers too. There's a lot of interest in measuring NAD levels. Currently the methods for measuring NAD levels are mostly found sort of in research institutions and used for research purposes. They're not really made sort of publicly available or as clinical or lab tests. I will say too, I think it's an interesting question. And I think one day we'll be able to maybe measure NAD levels and derive some meaning from that. But I think, with the current state of the science and what we know about NAD, I'm not sure that, as a field, we've really figured out enough about NAD and NAD levels to be able to, take somebody's NAD level measurements and really know what to do with that. And really what I mean by that is, we would expect lots of variation from one person to the next in terms of NAD levels, how much they exercise, what they're eating, different genetic predispositions could all affect sort of baseline NAD levels. And also sort of another interesting thing about NAD is that some of the enzymes involved in creating NAD are actually under control of the circadian clock. And so NAD levels aren't even necessarily constant throughout the day. So I think there are a lot of sort of just technical challenges that need to be worked out by the research community to really get to the point where we can measure somebody's NAD levels and get some actionable, useful information from that. So I'm optimistic, one day we'll get there, but I think right now we're a little bit ahead of where science is in that request.
All right. We have another question here. Are there any new human studies that are being published soon?
I believe so. I mean, there are certainly studies that are listed on clinicaltrials.gov that are listed as completed and are not yet published. So my hope is that scientists are busy writing up those manuscripts and getting them submitted so that we'll be able to see them soon. So I would estimate that we'll see at least a few new studies published this year.
And as the director of outreach, I'm going to put in a plug. If you go in and register on practitioner.truniagen.com, we do alert anybody who has registered there when new evidence is published. So we can help keep you abreast of what's going on with that regard. Okay, we have another question, and I probably should have tacked this one on too, kind of that whole testing question. What type of signs, symptoms, diagnoses would a practitioner look for to indicate that a patient might benefit from something like nicotinamide riboside or Tru Niagen?
Yeah, that's a great question too. And for me, I always go back to, what is, I think, the mechanism of NR and boosting NAD. And I think really at its heart, it's about helping improve mitochondrial health and supporting healthy cellular metabolism and energy production. So really anything kind of related to that would be a good reason to think about giving this to your patients. So, as I mentioned, a lot of consumers are interested in taking this because they maybe feel low energy and they want sort of a sustained energy boost. But, for your patients, there are all kinds of conditions, related to metabolic stress where, tissues are working harder than usual or, not keeping up with energetic demands, and, supporting mitochondrial health could really benefit those patients.
Liz, I've gotten a number of questions in related to kind of different diseases and conditions. Are there any customized dosing recommendations for different types of patients at this point?
Not yet. I wish there were, but I think right now, we need a little bit more of that clinical research to be published to really get a better picture of how NR is affecting these health outcomes in people and at what doses.
Okay. The other thing that I'll let folks know is on practitioner.truniagen.com and that NAD education hub, Liz and her team have done all of the hard work of pulling down the science around NR and NAD and categorizing it for you by specific condition to make it very easily searchable, because I've gotten a lot of questions around, is there any data in things like sarcopenia? And so that would be the best way to look and find some of that information.
Sure. This says, I've heard about getting NAD plus through IV. How is taking oral NR better?
Yeah, well, so there's, I think, a few different things to consider in that question. First of all, there are obviously some inherent risks in, taking anything via IV, versus orally as a supplement. So I think that's one thing to consider. And I think the other part of this is going back to kind of the difference between NR and NAD, and how cells can use those molecules. So, again, an NAD is a much larger molecule than NR. Contains lots of phosphate groups, is negatively charged, and cannot get into cells unless they have a transporter to help that molecule get inside of the cell. And so, I don't think a lot of cells, there's not a lot of evidence to suggest that NAD in circulation is able to get into cells. And so in order for it to actually be helping cells and tissues make an NAD, that NAD would presumably need to be broken down into smaller components to then enter cells and tissues and be converted back into NAD. And so I think that's sort of a general limitation with NAD as a supplement itself, whether that's through IV, whether that's through oral supplementation. And, in terms of just clinical research, there really hasn't been much done around NAD as a supplement in humans. So I think there's a stronger basis of evidence around NR currently in that arena.
Okay, some of the studies indicated dosage of 2,000 milligrams a day, and the label's 300. What would be the rationale for a practitioner that would influence increasing this dosage?
I mean, I think that would be kind of at your discretion based on why you are interested in giving this to your patients. Maybe if you have certain results or hopes in mind, you could always start at the lower dose and increase over time. I think that the higher doses that we've seen in some of the recent clinical studies are partly based on kind of coming up with human equivalent doses from some of the preclinical studies that they're trying to translate. To kind of address one of the earlier questions, I mean, one approach you could take while we wait for new human data to publish is to look at some of those preclinical studies. And if you find some preclinical evidence around your particular area of interest and see some outcomes that you would hope to see in your patients, that might give you a jumping off point to think about what maybe an equivalent dose might be in a human, to see if you could get the same kinds of results.
Great, thanks. Can you comment on taking NADH as a supplement?
Right. So I've seen both NAD plus and NADH as supplements. Both are, there's sort of limited clinical work done on either of those. I think if you go on clinicaltrials.gov, there's maybe a couple registered trials with NADH. I wouldn't expect there to be a big difference in those two molecules as supplements, based on them just being the reduced or oxidized form. The cellular environment is already pretty reducing, and so that's going to have some influences on those molecules and what form they actually take inside of the body, kind of regardless, I think, of what form you start with. So I think I would pretty much have the same thing to say about NADH as everything that I've said already about NAD+. I think there's questions about whether that molecule can actually enter cells. And so once it's in the body or even being absorbed into the bloodstream, my guess is that it's getting broken down to some extent into some of these smaller NAD precursors first.
Yeah, and I think the thing that I would add on to there that I've heard you talk about many times too is just also making sure that these particular nutrients, because of the hot nature of science and research in NAD, we see a lot of these supplements cropping up on the market, but we always want to make sure that we're checking that they have new dietary ingredient status too, to be having had a data package reviewed by FDA.
Yeah, no, that's a really great point. It's a very hot space right now. There's supplements cropping up all the time. And a lot of them don't get notified necessarily to the FDA. A lot of them don't have published clinical evidence to support their safety and efficacy at boosting NAD levels. So it's a really good thing to look out for.
Liz and Bria, thank you so much for taking the time to answer so many fantastic questions. It has been a pleasure talking about how NAD promotes metabolic resilience to support patient health. We apologize that to those who did not have their questions answered today, but we have run out of time. As mentioned before, we will follow up with you individually. If you have any additional questions, please feel free to contact our Natural Partners customer service team anytime.